excuse myself. No worries.
Okay, so we're going to kind of do some discussion working until like 1. And for part of this, because William is not a modeler, we thought it would be really, or not, sorry. William is more of a wet lab. So we thought it'd be really interesting to get his perspective on his work and also to get his perspective. So thanks, William.
Yes. So after some discussion with Morgan and Berta, then I thought I'd give a last minute talk. And I did give a talk yesterday across the street, and the reason being I need to visit my old lab and I'll become a division seminar. But so just brief introduce myself. I grew up in Taiwan and I moved to Toronto for my PhD study, and right across the street at the Princess Margaret Hospital. And then I moved to NIH, start as a postdoc, and then asked to stay as a senior staff. So as I stayed there almost 12 years. And then during the pandemic, tried to find a place to hide. So I moved to Little Rock, Arkansas, a small town. Very nice place for pandemic, but it really sucks after the pandemic is over. So there's only 200,000 people in that small town. So it's hard to do things and hard to hire people. But in any case, I want to show you today is for 2 purposes. One is to let you know what I've been doing so far. And second is I hope to stimulate some of the conversation for our upcoming discussion. So what I've been doing in a general picture is doing cancer immunotherapy with all different sort of spectrum. So one you might more familiar with the checkpoint blockade immunotherapy. It starts with monotherapy or combination therapy. And they also have that combined with surgeries called adjuvant therapy or neoadjuvant therapy, depends on the timing. We're also working on mostly T-cell therapy. So the joke is we're doing everything other than CAR-T. Everybody knows CAR-T, and we're just not doing CAR-T, even though we are the second group that get the FDA approval for the CAR-T therapy. And so basically what happened is we have the peripheral blood, and when gene engineer put in the artificial TCR gene or the CAR-T gene into that, expand and transfer to the patient. We could also grow the T cell from the tumor and then culture in vitro and then transfer back to the patient. And now we all have one example that's been approved by FDA for treatment this year. We also partially working on neoantigen vaccine. Neoantigen means that it's I'll explain more later. It's the tumor generates some mutation, and that can be recognized by immune system, especially T cell, and then we use that to generate the vaccine which for the patient treatment. So just to clarify a little bit, so there are 2 types of tumor antigen. One is called tumor-associated antigen. That means they are the normal gene, but they overexpress in tumor, and they can be recognized by T cell. And the other type that we are focusing today is some of the tumor, they have mutation, and mutation generate different protein, and that can be recognized by T cell, and we call them tumor-specific antigen or call it neoantigen. So it's again what you are doing now. It's at the beginning that we use identify the whole exome sequencing to identify the mutation, and using the in silico analysis to predict the binding of the peptide. So the basic immunology is the antigen will be processed and present on the cell surface by MSC, and in the human we call HLA. But there are different diversity of the HLA, and there are some common one and uncommon one, and then you can use the prediction or the neural network to train them to predict what kind of peptide that can bind to the surface, and those peptides by chance that will be recognized by T cell. So then we screen those candidate peptide and use that to identify the T cell that recognize those peptides. So in my work as a postdoc, we try to get rid of that. So we still doing that, but we have the combination of the mutation in the middle and then put in all the amino acid on the side and make it as a mini gene. And then we congregate 6 to 12 mini gene together. We call it tandem mini gene. And then we screen those tandem mini gene, which can include all the combination of the different HLA type, which we will get rid of the in silico analysis. And then using that, we can identify the antigen and then use that for our research purpose and for treatment. So after 10 years old, this is the top line clinical trial that was done by Moderna initially. They cooperate with us. And as you may know, that during the COVID, most of the mRNA vaccine has been used for the production for COVID. And now the pandemic is over, so they shift back for the production to do something else. So this is one of the top things that they are working on, including also for other companies such as Biovance and others. And so the clinical trial that's published early this year is phase 2B clinical trial. It compared the melanoma patient treated with PD-1 or the PD-1 plus the mRNA vaccine. So the messenger RNA vaccine that's encoding individualized neoantigen, which means that every patient come in, they will perform the whole exome sequencing, identify the mutation, usually around 200 to 300 mutations, and they select some of them, put them into the tandem mini construct, and then make it become the RNA and use that as a vaccine. And then they compare that, you see there's some improvement compared to the PD-1 alone. So now they are in the phase 3 clinical trial, and hope that we'll see that coming to the market very soon. So I'm grateful that I took the credit for that. That's all based on the contract that I did 10 years ago. Yeah. So as a research side, want to do more than just what we have now. So one of the things that I've been working on, we could call it the Moonshot project, is most of the mutation that's identified by the whole exome sequencing is based on the single nucleotide variant, such as one point mutation from one amino acid to the other. But they could also be anything that's different can be recognized by T cell and immune system. It's either insertion, deletion, fusion, or some alternative splicing. Even some phosphorylation can be recognized by T cell. But we want to know whether those mitochondrial DNA mutation, which also generate protein, and whether they can become neoantigen or not. So just very briefly say the mitochondrial DNA is totally different species. They have their own DNA and they have certain gene which very packed, there's no intron, and they also have tRNA and ribosomal RNA and their own coding system. So it's a huge learning curve to switch to learning how to deal with the mitochondrial DNA. So we just submit the paper. So how we have is we have an approach. We first acquire all the natural occurred tumor mutation that's detected in the mitochondrial DNA. So it's about 3,800, and then we're focusing on the single point mutation. So again, we start and with normal amino acid at each site, and then because it's hard to do all the tandem mini gene at once, so we decided to do the prediction algorithm, which is most common one is HLA-A2, and then we synthesize the top 300 candidate, and then so at this point that we're still using some prediction, and then we start the generate the T cell population we start, and then once they have the candidate, we perform the single cell analysis and then do the validation. So after screening 10 patients with 10 peptide pool, we only got one hit. And then here's the validation for that one hit. So and then what we did is the single cell analysis. So we have the T cell population, we stimulate them, and then we get two different type of data. One is the single cell transcriptome, the RNA expression data, and the other one is the TCR sequence. So we use those TCR sequence to identify new TCR. So here's a single cell analysis clustering, and in the previous study, we found that the interferon gamma and IL-2 are two base markers. So it's concentrated on cluster 12 and 14. So we can also plot them in the regular dot plot, and then for each individual dot, it represents individual single cell, and then we ask a question: what's the TCR expressed in those T cells? And then we totally identify 4 of them. We label them with A, B, and C, and D, and some of them they are not detectable, so we put U as a label, and or some of them they are non-specific, put it on the N as a label.
For the graphs, sorry, can you explain the y-axis on the these are the sequences? Like
sorry, I speak too fast. So the yeah, thank you. The y-axis is the UMI, unique molecular identifier. It's basically the expression label out in the interferon gamma. Okay. It's the in the single cell world that they use UMI instead of the other way. Sometimes I like how many copies of RNA that they use UMI. Yeah. So it just plot with the intensity of interferon gamma and IL-2. But while the key message is we study every single cell that was high interferon gamma, and then we ask the question what's the TCR is in that single cell. And then we did the validation. The validation means that we take out the sequence, which is totally digitalized, and then we send to a company and in China, and they will make it convert to the real scene, so the synthesize a nucleotide and put that into a retrovirus contrast, and then we re-express that in the healthy T cell. And I don't want to explain more, but it basically means that it recognizes the mutation but not the wild type. And then we also did that with the tumor cell with transfer with the mutation is also get recognized by those T cell but not the wild type. So yeah, I will skip some of the slides. And so here's something that the key message I have to bring up today. It's I changed the title. How can we drive for the next step? So what we did before, and this is not the first time, but you can we have 4 TCR, A, B, C, and D, and then is non-specific. So we found out the non-specific one is all focused on cluster 12, and the specific one concentrate on cluster 11 and 14. Sorry, that is too small. But the one say is the cluster 11 is so-called exhaust T cell, means that those T cell has been stimulated and they are exhausted, they cannot be stimulated again, so we couldn't find good marker for that. But when you look at the cluster 14, which is, you can call it reverse gating, or you can call it the technique for clustering, you can always ask what other gene that's also expressed in that cluster. So it's a long list, 100 gene, but we'll pick out a few that might be useful. One is the TMFF9, it's 4-1BB ligand. The other one is IL-10. So then, since we have a new gene, so then we ask, and you can see that it's highly expressed in that single cluster, but not the others. So then we ask the question, reverse way, can we use those 2 new markers to identify the new TCR or the old one? And the answer, yes, all the previous identified TCR that can be identified by those 2 gene markers, and we will also identify 2 new TCR, but we need to do more tests for that. So yeah, I just want to show you as an example that as a web bench scientist, that we also doing analysis, and the analysis help to generate new hypotheses and also the new research direction. So the other thing that's okay, I can talk a little bit, so because I may need your opinion on that. So getting into a very crazy stuff. So mitochondrial DNA, it's complicated because, as you may appreciate, each single cell they have from 200 to 1,000 copy of mitochondria. and each one have individual sequence. So just put that into perspective that those point mutation is just very diverse, and every time we study every single one, and it could be 50%, could be 20%, or could be 0%. And the problem being when they divide into the daughter cell, it's not always copy the original one. Somehow when the cell divide, it could be unequally divide. So it could be 100%, could be 0%. And we don't know how to deal with that. And so about 2-3 years ago, we developed our own technique, but the other people also developed new technique, and we end up using theirs. So it's they use the single cell, but it fixes the cell, so the mitochondria will stick to the cell, and then we can use the attack C, which is will attack the chromosome that's opening part. So we use that to know that where the gene has been expressed. On the other hand, because the mitochondrial DNA is not being protected, so we can check those mitochondrial DNA to small pieces and sequence them, and then we can analyze both of the data together, so we know where to those mutation that's been located. So let's see. So what we did that is for the tumor cells, and we dissociate tumor cell and purify them by a marker EpCAM1, and then perform the single cell analysis. And unfortunately, or fortunately, that we got two big clusters. One is a tumor cluster, the other one is macrophage cluster. For some reason, that the macrophage also have some low level EpCAM1 expression, but we're pretty sure they are the macrophage because of the gene marker for EpCAM1 is mostly on tumor, but the CD40 and TREM1 is on the macrophage cluster. And then we can also plot the same thing for the mutation. So we only work on the mutation that changes amino acid sequence. So we can see that this one, almost 99% of the tumor cell have that mutation. And this one is only partial one. And then this one is expressed everywhere. We use that as a negative control. Here's the heat map showing that most of the tumor cell have that mutation. This is important for us because we want to target a mutation that is present on all tumor cells, so the tumor cell get killed. If it's only partial, like the second one, then some of the tumor cell get killed, and the rest of them eventually grow back out, and so make the treatment ineffective. And we also see some macrophage got some sticky DNA, or they just taking out some of the tumor cell. There's some signal there, but the PBMC, there's no signal. So that's very tumor specific. And so even crazier now I'm doing now is we also doing that for T cells. So we generate some new hypotheses thinking that T cell may be mitochondrial DNA damage also may get involved in T cell. So I just want to skip most of the slide. What I want to show is so for example, the T cell being treated with cisplatin, we could detect quite a lot of mutation here and here, and then we are working hard to find the clustering that's associated with those mutations, and then using the same approach to generate new hypotheses. And yeah, I'll skip most of the slide. So why I'm in trouble now is we know that we have the T cell, once they treat with chemotherapy drug, it could induce some mutation. But some of them, as expected, they induce negative effect, and then some of them could see it, some of them they just die, and we cannot see it. So that's normal part. But we also see a lot of them maybe passenger, or there's no effect for that, and we detect those mutation, but without any physiological difference. But they also have somehow positive effect, and that actually increase the survival for T cell. At the same time, there's a Nature paper published that they use experimental way to test the hypothesis, and they can show that the the current mutation depends on the environment. So what I need you guys help is I don't know how to deal with this complicated system. Yeah, as you know, that as a T cell progress, the T cell divide, and then but they also carry on mutations. Some mutations they have negative effects, some they have positive effect, and then you have different results. So it's open. So yeah, so that's all I have. So so most of the work been done by the MD-PhD student Catherine Kirkpatrick, and some were done by postdoc Shuang. So yeah, thank you. Yes, please. Yes, please go first.
Okay, I have a lot of so I was very curious about when you were doing the validation experiments for the T cells, right? When you were looking, and so you were looking at 4-1BB as a marker, and then IL-10.
Yeah.
Can you give us some biological context as to why these two particular markers you were looking at? So the 4-1BB. Specifically, is that a marker which is indicating exhaustion for these cells? Like, how are you guys thinking about? Do you know which one? I don't remember the name of the slide or the number of the slide. So I was just really curious about how you guys, because you can look at a lot of things, right? For
that's the point. Yes, you get it.
So what was the rationale for?
The rationale is the counterintuitive. Yeah. So of course we look at the top 100 gene,
right.
Look at whatever that's familiar. Right. And other than the one we know, interferon gamma, that's obviously when T cell recognize and you activate interferon gamma and IL-2, and those obvious one we test and they're known. And these two are actually quite new. So everybody know 4-1BB because 4-1BB is the one that also get activate another ligand.
Right.
And IL-10
IL-10 is complicated.
It's also complicated. Some people say it's about T helper 1, T helper 2, and the T helper 1 is associated with interferon gamma, and T helper 2 associated with IL-4, and IL-10 get involved. So that's how I feel. We can look at those two genes because it's new, because nobody expect that would.
I see that they're a little counterintuitive, and I think that's where thinking about modeling, kind of thinking through some of these counterintuitive sort of conclusions could be really helpful. So yeah, thank you for that. And then the second question I had was like the mitochondrial DNA point mutations. Fascinating. These are very tumor specific or T cell specific mutations that you guys are after. So for the tumor specific ones right now, lots of people are really excited about ctDNA, right? Which is sort of, mutations that would pick up from circulation. So are these types of mitochondrial mutations the ones that could be detected in ctDNA?
Ha, 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 ha. You just get a new project. So it's a long trend. It's at the very end of my career at NIH, I was asking a question whether we can detect genomic DNA mutation at a single cell level, and I failed terribly because it's so hard to do that. Because imagine you only have 2 copies.